TB Talks Education. Let us proceed with biology. How mutation causes variation in organisms. So now, all along, I have been saying that mutation refers to change in amount, structure, and arrangement of DNA in an organism. So, mutation refers to change in amount, structure, or arrangement of DNA in an organism. Change in amount, that is one. Structure or arrangement of DNA in an organism or in a living thing. So now, these changes here cause variations in genotypes of organisms. So these changes cause variations. These changes cause variations in the genotypes. In the genotypes of organisms, and thus the characteristics or phenotypes of organisms, also change. So these changes cause variations in the genotypes of organisms, and thus the characteristics or phenotypes of organisms also change. So now, here, if I can illustrate with an example whereby inheritance of sickle cell anemia. So a particular example here is the inheritance of sickle cell anemia, whereby a mutation occurs, a gene mutation, also called point mutation, occurs in the genotype or DNA, and one gene, one triplet, or one base within the triplet is replaced by another one that now changes the gene which codes for amino acid glutamine. And now they change the triplet or the mutant triplet or mutant gene codes for base valine. And so when base valine replaces glutamine at position number 6 of the beta chains of hemoglobin, that change leads to a change of the normal hemoglobin pigment to an abnormal hemoglobin pigment, which is a mutant called hemoglobin S. And now that abnormal hemoglobin now causes the red blood cells to change from the normal biconcave disc shapes to the sickle shape, what we call sickle cell anemia. So here I can write that For example, a gene or point mutation, occurs in the triplet bases, in the triplet of bases on DNA strand. The gene or the triplet codes for amino acid glutamine. So what happens is that the mutant triplet now codes for amino acid valine. So now here, in brief, replacement of glutamine by valine. Replacement of glutamine by valine in the beta chains, in the beta chains of hemoglobin, leads to formation, or leads to a mutant hemoglobin. Leads to a mutant hemoglobin called hemoglobin S. This results into change of the normal bicongave disc shape of red blood cells. Okay? The hemoglobin S, which is the mutant hemoglobin, results into change of the normal bicongave disc shape of red blood cells to sickle shape, resulting in a genetic disease called sickle cell anemia. So, what's the normal bicondyl disc shape of the red blood cells changes to sickle shape, then we say the individual has sickle cell anemia. However, to finalize with, individuals who are heterozygotes have an advantage because they are naturally resistant to malaria. However, heterozygotes, those are individuals with normal hemoglobin and abnormal hemoglobin. That is HbA, HbS. HbA is the normal hemoglobin and the HbS is the abnormal hemoglobin. Have heterozygote advantage, or these ones enjoy heterozygote advantage since they are naturally resistant to malaria, and are called the traits, So individuals who have what we call sickle cell trait are the heterozygotes. They are different from the individuals with sickle cell anemia. And now, such individuals have part of their hemoglobin being normal and the other half is abnormal. Of course, it is hemoglobin S. Now, those individuals are not easily attacked by the Plasmodium parasite that causes malaria, and because of that, they are naturally resistant to malaria. And those individuals are called the traits, or the individuals with sickle cell trait. Dear scholars, that is how mutation causes variation in organisms. Let us meet some other time with continuation on mutation.